Welcome back to Bioscholar. Have you ever wondered where we get the energy to do our work and how this energy is produced? Let's find out in this video. To perform our daily tasks, we need energy, which we get from the food we eat. Food contains nutrients that provide energy in the form of ATP. Carbohydrates are our primary source of energy. But how exactly do these carbs produce energy, and how much energy is produced? Let's break it down step by step. While proteins and fats also contribute to energy production, we'll focus on carbohydrate metabolism for now and cover proteins and fats in upcoming videos. Carbohydrates are polymers of glucose. When broken down, they yield glucose monomers. These glucose monomers undergo a process called glycolysis, producing pyruvate. Pyruvate then enters the Krebs cycle, generating electron carriers. These electron carriers proceed to oxidative phosphorylation, ultimately producing energy in the form of ATP. This entire process is known as cellular respiration. In essence, cellular respiration is the process of producing energy by metabolizing food nutrients. Proteins and fats can also produce pyruvate, which then enters the Krebs cycle, following the same pathway as carbohydrates. Now, let's dive into this and explore how ATP is produced. Glycolysis comes from the Greek words glyco and lysis, where glyco means sugar and lysis means splitting. Thus, glycolysis refers to the splitting of sugar. This process breaks down the sugar we consume into energy, supporting various cellular processes. Formally, glycolysis is defined as the metabolic pathway that converts glucose into pyruvate, generating small amounts of ATP and NADH in the process. Glycolysis is a series of 10 enzyme-catalyzed reactions. It is the first step in cellular respiration and fermentation. This process is anaerobic, meaning it does not require oxygen. It takes place in the cytoplasm of both prokaryotic and eukaryotic cells. Before we dive into the steps of glycolysis, let's talk about what we need to get started. Glycolysis is a series of reactions, and at each stage, different molecules play crucial roles. First off, we need a substrate, and in this case, it's glucose, the simple sugar we get from our food. Next, we require various enzymes. These enzymes act like little helpers that speed up each step of the process, ensuring everything runs smoothly. We also need energy carriers. These molecules, like ATP and NAD, are essential for providing the energy needed to drive the reactions forward. And of course, all of this takes place within the cell. Specifically, glycolysis happens in the cytoplasm, which is the fluid-filled area inside the cell. So, to recap, for glycolysis to occur, we need glucose, enzymes, energy carriers, and the cytoplasm of the cell. Now, let's move towards the process. As we go through the steps, notice how many ATP molecules are used and how many are produced at the end. In the first step, an enzyme named hexokinase phosphorylates glucose to trap it in the cell and make it more reactive. A phosphate group is added to glucose, converting it into glucose 6-phosphate. This is called glucose 6-phosphate because the phosphate group is attached to carbon number 6 of glucose. But where does this phosphate group come from? It's donated by an ATP molecule. The ATP molecule is broken down into ADP and a phosphate group. This phosphate group is then attached to glucose, turning it into glucose 6-phosphate. To summarize, glucose plus ATP is converted into glucose 6-phosphate and ADP. This reaction is catalyzed by the enzyme hexokinase. In the second step of glycolysis, the glucose 6-phosphate produced in the first step undergoes a structural rearrangement to form fructose 6-phosphate. This reaction is catalyzed by the enzyme phosphoglucose isomerase. This isomerization step is important because it converts the 6-membered ring of glucose 6-phosphate into the 5-membered ring of fructose 6-phosphate, making the molecule more suitable for subsequent steps in glycolysis. At the end of this step, we obtain fructose 6-phosphate, an isomer of glucose 6-phosphate, catalyzed by phosphoglucose isomerase. In the third step of glycolysis, fructose 6-phosphate is phosphorylated to form fructose 1,6-bisphosphate. This reaction is catalyzed by the enzyme phosphofructokinase 1, which is one of the key regulatory enzymes in glycolysis. Phosphofructokinase 1 is an allosteric enzyme, meaning its activity can be regulated by other molecules. It is activated by AMP, which signals low energy levels in the cell, and inhibited by ATP and citrate, which signal high energy levels. This regulation ensures that glycolysis proceeds when the cell needs energy and slows down when the energy supply is sufficient. In this step, a second ATP molecule is used to add a phosphate group to fructose 6-phosphate, converting it into fructose 1,6-bisphosphate. In this molecule, phosphate groups are attached to carbon 1 and carbon number 6. This phosphorylation is crucial as it further activates the sugar molecule, preparing it for the subsequent steps in glycolysis. To summarize, fructose 6-phosphate plus ATP is converted into fructose 1,6-bisphosphate and ADP. This reaction is catalyzed by PFK1, a key regulatory enzyme in glycolysis. In the fourth step, the 6-carbon molecule fructose 1,6-bisphosphate is split into two 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This reaction is catalyzed by the enzyme aldolase. Aldolase performs this cleavage by breaking the bond between the third and fourth carbon atoms of fructose 1,6-bisphosphate. This step is important because it produces two molecules that can be further metabolized in the subsequent steps of glycolysis. Even though two 3-carbon molecules are produced, only glyceraldehyde 3-phosphate continues directly in the glycolytic pathway. The other product, dihydroxyacetone phosphate, is quickly converted into G3P by the enzyme triose phosphate isomerase, ensuring that both molecules produced in this step can be utilized in the pathway. At the end of this stage, we obtain two 3-carbon molecules, and this reaction is catalyzed by aldolase. In the sixth step of glycolysis, glyceraldehyde 3-phosphate is oxidized and phosphorylated to form 1,3-bisphosphoglycerate. The enzyme glyceraldehyde 3-phosphate dehydrogenase catalyzes this reaction. During this step, NAD is reduced to NADH, capturing energy in the form of high-energy electrons. Additionally, an inorganic phosphate is added to G3P without the use of ATP, forming 1,3-bisphosphoglycerate. This step is crucial as it generates NADH, which can be used in oxidative phosphorylation to produce more ATP. To summarize, glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate while reducing NAD to NADH. This reaction is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. In the next step, 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. This reaction is catalyzed by the enzyme phosphoglycerate kinase. During this step, a phosphate group from 1,3-bisphosphoglycerate is transferred to ADP to form ATP. This is an example of substrate-level phosphorylation, where a phosphate group is directly transferred from a substrate molecule to ADP, forming ATP. This step produces two molecules of ATP for each glucose molecule because each glucose molecule generates two molecules of 1,3-bisphosphoglycerate. At the end of this step, we obtain two 3-phosphoglycerates while generating ATP molecules, which is catalyzed by phosphoglycerate kinase. In the eighth step of glycolysis, 3-phosphoglycerate is converted into 2-phosphoglycerate. This reaction is catalyzed by the enzyme phosphoglycerate mutase. Phosphoglycerate mutase catalyzes the transfer of a phosphate group from the third carbon to the second carbon of the molecule. This enzyme operates via a mechanism that temporarily adds a second phosphate to the molecule, creating an intermediate called 2,3-bisphosphoglycerate. The enzyme then removes the original phosphate from the third carbon, resulting in the formation of 2-phosphoglycerate. This step is crucial for preparing the molecule for the subsequent dehydration reaction, generating a high-energy compound necessary for ATP production in the next step. At the end of this stage, 3-phosphoglycerate is converted into 2-phosphoglycerate through the action of phosphoglycerate mutase, involving an intermediate formation of 2,3-bisphosphoglycerate. In the 9th step of glycolysis, 2-phosphoglycerate is converted into phosphanol pyruvate. The enzyme enolase catalyzes this reaction. During this step, enolase removes a molecule of water from 2-phosphoglycerate, resulting in the formation of phosphanol pyruvate. Phosphanol pyruvate is a high-energy compound, with a high phosphoryl transfer potential, which is essential for the next step in glycolysis. So, in the end, 2-phosphoglycerate is converted into phosphanol pyruvate through the removal of a water molecule. This reaction is catalyzed by enolase. In the 10th step of glycolysis, phosphanol pyruvate is converted into pyruvate. The enzyme pyruvate kinase catalyzes this reaction. During this step, the high-energy phosphate group from PEP is transferred to ADP, forming ATP. This is another example of substrate-level phosphorylation. The result is the production of pyruvate, which is the end product of glycolysis. This step is crucial as it not only produces ATP, but also forms pyruvate, which can enter the mitochondria for further energy production through the citric acid cycle and oxidative phosphorylation under aerobic conditions, or be converted into lactate under anaerobic conditions. So, finally, in the end, we obtain pyruvate while generating ATP. This reaction is catalyzed by pyruvate kinase. The first part of cellular respiration ends here. Let's examine what is used and produced during the process. Summarizing the energy yield, 4 ATP molecules are produced, but since we used 2 ATP molecules initially, the net gain from glycolysis is 2 ATP molecules. Additionally, 2 NADH molecules are produced during glycolysis. The Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid cycle, is named after Hans Krebs, who identified its steps in the 1930s. Hans Krebs first identified the cycle in 1937, earning the Nobel Prize in Physiology or Medicine in 1953. It is a series of chemical reactions used by all aerobic organisms to generate energy through the oxidation of acetyl-CoA derived from carbohydrates, fats, and proteins. This cycle is a crucial part of cellular respiration that takes place in the mitochondria, where enzymes facilitating the reactions are located. It involves the oxidation of acetyl-CoA to carbon dioxide and water, producing energy in the form of ATP and electron carriers NADH and FADH2, which enter the electron transport chain. The Krebs cycle occurs in the matrix of the mitochondria in eukaryotic cells and in the cytoplasm in prokaryotes, playing a key role in the cell's energy production and metabolic processes. Before proceeding to the steps of the Krebs cycle, let's see what is required for the process. Acetyl-CoA, derived from the metabolism of carbohydrates, fats, and proteins, is essential. We also need NAD and FAD, which are electron carriers that get reduced to NADH and FADH2. Additionally, ADP and inorganic phosphate are needed for ATP synthesis. Finally, water is used in various reactions throughout the cycle. So to recap, to Krebs cycle, acetyl-CoA, electrons carriers, ADP, inorganic phosphate, and water are needed. Besides all of these, we need a cellular environment which is obviously the mitochondrial matrix. Let's discuss the steps of the Krebs cycle. To start the process, we require acetyl-CoA. As I mentioned, the Krebs cycle is the second step of cellular respiration, so technically, the end product of the first step should enter into the second step. The end product of the first step is pyruvate. The process starts with acetyl-CoA. This means pyruvate has undergone some transformation. This reaction is called pyruvate decarboxylation. So, the first step is the conversion of pyruvate into acetyl-CoA. Pyruvate decarboxylation is a crucial biochemical reaction that connects glycolysis and the Krebs cycle in cellular respiration. This process converts pyruvate, the end product of glycolysis, into acetyl-CoA, which then enters the Krebs cycle. This reaction is catalyzed by the pyruvate dehydrogenase complex, a large multi-enzyme complex made up of these three enzymes. Several cofactors and coenzymes are required for the pyruvate dehydrogenase complex to function properly. During this process, pyruvate loses one carbon as carbon dioxide. The remaining 2-carbon acetyl group is then transferred to coenzyme A to form acetyl-CoA. This reaction is catalyzed by the pyruvate dehydrogenase complex and also produces NADH from NAD. The overall reaction can be summarized as follows: pyruvate plus CoA plus NAD plus yields acetyl-CoA, carbon dioxide, NADH, and a hydrogen ion. Now that acetyl-CoA is ready, it enters the Krebs cycle. In the first step of the Krebs cycle, acetyl-CoA combines with a 4-carbon molecule called oxaloacetate to form a 6-carbon molecule called citrate. This reaction is catalyzed by the enzyme citrate synthase. During this step, the acetyl group from acetyl-CoA is transferred to oxaloacetate, completing the formation of citrate. This marks the beginning of the series of reactions that will lead to the production of energy and electron carriers in the Krebs cycle. In the second step of the Krebs cycle, citrate undergoes a rearrangement to form isocitrate. This reaction is catalyzed by the enzyme aconitase. Citrate is first converted to an intermediate called cis-aconitate through the removal of a water molecule. Then, water is added back to this intermediate to form isocitrate. This step is crucial as it prepares the molecule for the subsequent oxidative decarboxylation. In the third step of the Krebs cycle, isocitrate is oxidized and decarboxylated to form alpha-ketoglutarate. This reaction is catalyzed by the enzyme isocitrate dehydrogenase. During this step, isocitrate is oxidized, which converts NAD+ to NADH. The process also releases a molecule of carbon dioxide, resulting in the formation of alpha-ketoglutarate. This step is important as it generates NADH, which will be used in the electron transport chain to produce ATP. In the fourth step of the Krebs cycle, alpha-ketoglutarate is further oxidized and decarboxylated to form succinyl-CoA. This reaction is catalyzed by the alpha-ketoglutarate dehydrogenase complex. During this step, another molecule of carbon dioxide is released, and NAD+ is reduced to NADH. Additionally, coenzyme A is added to the remaining 4-carbon molecule to form succinyl-CoA. This step is significant because it generates another NADH molecule and prepares the substrate for subsequent reactions in the cycle. In the fifth step of the Krebs cycle, succinyl-CoA is converted into succinate. This reaction is catalyzed by the enzyme succinyl-CoA synthetase. During this step, the CoA group is released from succinyl-CoA, and this release provides the energy needed to synthesize a molecule of guanosine triphosphate from guanosine diphosphate and inorganic phosphate. In some cells, GTP is then used to produce ATP. This step is significant because it directly produces a molecule of GTP or ATP, which is used as an energy source by the cell. In the sixth step of the Krebs cycle, succinate is oxidized to fumarate. This reaction is catalyzed by the enzyme succinate dehydrogenase. During this process, 2 hydrogen atoms are removed from succinate and transferred to the electron carrier flavin adenine dinucleotide, reducing it to FADH2. This step is unique because succinate dehydrogenase is embedded in the inner mitochondrial membrane and also participates in the electron transport chain. The production of FADH2 is important as it will be used in the electron transport chain to generate ATP. In the seventh step of the Krebs cycle, fumarate is hydrated to form malate. This reaction is catalyzed by the enzyme fumarase. During this process, a water molecule is added to fumarate, converting it into malate. This step is essential as it prepares the substrate for the final oxidation reaction in the Krebs cycle. In the eighth and final step of the Krebs cycle, malate is oxidized to regenerate oxaloacetate. This reaction is catalyzed by the enzyme malate dehydrogenase. During this process, Malate is oxidized. NAD+ is reduced to NADH. The regeneration of oxaloacetate is crucial as it allows the Krebs cycle to continue by providing the starting molecule that combines with acetyl-CoA in the first step. This step also produces another NADH molecule, which will be used in the electron transport chain to generate ATP. Let's summarize the energy production from the Krebs cycle and the linking reaction. The Krebs cycle produces 3 molecules of NADH, 1 molecule of GTP which is equivalent to ATP, and 1 molecule of FADH2 per cycle. Additionally, the linking reaction produces 1 NADH. So, together, the linking reaction and Krebs cycle produce 4 NADH. Since glycolysis produces 2 pyruvates, this means 2 Krebs cycles occur, resulting in 8 NADH, 2 GTP or ATP, and 2 FADH2 molecules. In total, for the first 2 stages of cellular respiration, we have collected 12 electron carriers, 10 NADH and 2 FADH2 molecules. These molecules then enter the electron transport chain to yield further energy. The third stage of cellular respiration, oxidative phosphorylation, is the process of ATP synthesis driven by the transfer of electrons from electron donors such as NADH and FADH2 to electron acceptors, primarily oxygen, via redox reactions. This process consists of 2 main parts: electron transport chain and chemiosmosis. Let's see who introduced this concept. In 1939, Volodymyr Belitsa coined the term oxidative phosphorylation, establishing the concept of ATP production through redox reactions and phosphorylation. Then in 1949, Maurice Friedkin and Albert demonstrated that the coenzyme NADH links metabolic pathways, such as the citric acid cycle, to ATP synthesis. They showed that NADH, generated in the citric acid cycle, donates electrons to the electron transport chain, driving oxidative phosphorylation. Later in 1961, Peter Mitchell proposed the chemiosmotic theory, explaining the mechanism of ATP production during oxidative phosphorylation. He suggested that the electron transport chain creates a proton gradient by pumping protons across the inner mitochondrial membrane. Mitchell explained that ATP is synthesized as protons flow back into the mitochondrial matrix through ATP synthase, driven by the proton motive force. This revolutionary idea earned him the Nobel Prize in Chemistry in 1978. Now let's have a look where this process is occurring in the cell. In eukaryotic cells, the electron transport chain is located in the inner mitochondrial membrane. In prokaryotic cells, such as bacteria, it is located in the plasma membrane. Prokaryotic cells, unlike eukaryotic cells, lack internal membrane-bound organelles, therefore, their electron transport chain is located in the plasma membrane. So, as I mentioned earlier, oxidative phosphorylation consists of 2 parts, the electron transport chain and chemiosmosis. The electron transport chain creates a proton gradient, and chemiosmosis uses this gradient to synthesize ATP. The electron transport chain consists of 4 main protein complexes and 2 mobile carriers, U-biquinone, also known as coenzyme Q, and cytochrome C. Electrons are transferred through these complexes, leading to the pumping of protons from the mitochondrial matrix into the intermembrane space, creating an electrochemical gradient. In chemiosmosis, protons flow back through ATP synthase, driving ATP production. Before moving on to the detailed process, let's see what is required and used in the process. First things first, we need electron donors, NADH and FADH2. They are produced during glycolysis, the citric acid cycle, and fatty acid oxidation. Next, we need electron acceptors, oxygen, which is the final electron acceptor and gets reduced to water. Then, we need protein complexes and mobile carriers. Finally, we need ADP and inorganic phosphate for the synthesis of ATP, and the cellular environment, which is the mitochondria. This process incorporates the mitochondrial matrix, inner membrane, and intermembrane space. Now that we have established that the electron transport chain pumps protons from the mitochondrial matrix into the intermembrane space, and that chemiosmosis drives these protons back into the mitochondrial matrix to generate ATP, let's see how the process works in detail. Here are the names of the complexes used in oxidative phosphorylation. Now, let's dive into the first stage of the electron transport chain, focusing on complex 1, also known as NADH dehydrogenase. NADH dehydrogenase is responsible for transferring electrons from NADH to coenzyme Q, or ubiquinone, and for pumping protons from the mitochondrial matrix into the intermembrane space. Here's how it works. First, NADH, which is produced during glycolysis, the citric acid cycle, and fatty acid oxidation, arrives at complex 1. NADH is oxidized, releasing 2 high-energy electrons and a proton. These 2 electrons are transferred to flavin mononucleotide, or FMN, a prosthetic group in complex 1, reducing it to FMNH2. Next, the electrons are passed through a series of iron-sulfur clusters within complex 1. These clusters facilitate the sequential transfer of electrons to ubiquinone. Finally, the electrons are transferred to ubiquinone, reducing it to ubiquinol. The energy released during the transfer of electrons through complex 1 is used to pump 4 protons from the mitochondrial matrix into the intermembrane space. This action contributes to the creation of the proton gradient. To summarize the first stage, electrons transfer from NADH to FMN, then through iron-sulfur clusters, and finally to ubiquinone, reducing it to ubiquinol. This process results in the pumping of 4 protons from the matrix to the intermembrane space, contributing to the proton gradient. Now, let's move on to the second stage of the electron transport chain, focusing on complex 2, also known as succinate dehydrogenase. Succinate dehydrogenase is responsible for transferring electrons from succinate, via FADH2, to coenzyme Q, or ubiquinone. Here's how it works. First, succinate, a substrate from the citric acid cycle, is oxidized to fumarate within complex 2. This reaction reduces the FAD prosthetic group to FADH2. Next, the electrons from FADH2 are transferred to a series of iron-sulfur clusters within complex 2. Like the first stage, these clusters facilitate the transfer of electrons to ubiquinone. Finally, the electrons are transferred to ubiquinone, reducing it to ubiquinol. However, it's important to note that, unlike complex 1, complex 2 does not pump protons across the membrane. Complex 2 contributes to the electron transport chain by supplying electrons from FADH2 to ubiquinone without contributing directly to the proton gradient. This ensures the continued flow of electrons through the chain, supporting the overall process of oxidative phosphorylation. To summarize the second stage, electrons transfer from succinate, through FADH2 and iron-sulfur clusters, to ubiquinone, reducing it to ubiquinol. Although complex 2 does not pump protons, it plays a crucial role in maintaining the flow of electrons through the electron transport chain. Now, let's move on to the next stage of the electron transport chain, focusing on complex 3, also known as the cytochrome B and C1 complex. Cytochrome B-C1 complex is responsible for transferring electrons from ubiquinol to cytochrome C and for pumping protons from the mitochondrial matrix into the intermembrane space. First, ubiquinol is oxidized at complex 3, releasing 2 electrons and 2 protons. The electrons then follow 2 separate pathways. One electron is transferred to the Re-Ski iron-sulfur protein and then to cytochrome C1, eventually reaching cytochrome C. The other electron is transferred to cytochrome B and then to another molecule of ubiquinone, forming a semiquinone radical. During this process, cytochrome B-C1 complex pumps 4 protons from the mitochondrial matrix into the intermembrane space, contributing to the proton gradient. Cytochrome C, a small heme protein, then accepts electrons from complex 3 and carries them to complex 4. To summarize the third stage, electrons transfer from ubiquinol to cytochrome C through the cytochrome BC1 complex. This process results in the pumping of 4 protons from the matrix to the intermembrane space, enhancing the proton gradient. Now, let's move on to the fourth and final stage of the electron transport chain, focusing on complex 4, also known as cytochrome C oxidase. Cytochrome C oxidase is responsible for transferring electrons from cytochrome C to oxygen, the final electron acceptor, and for pumping protons from the mitochondrial matrix into the intermembrane space. First, cytochrome C carries electrons to complex 4, with each cytochrome C molecule donating 1 electron. The electrons are then transferred through a series of copper and heme groups within complex 4, and finally accepted by oxygen. 4 electrons are required to reduce 1 molecule of oxygen to 2 molecules of water. During this process, complex 4 pumps protons from the mitochondrial matrix into the intermembrane space, contributing to the proton gradient. To summarize the final stage, electrons transfer from cytochrome C through complex 4 to oxygen, reducing it to water. This process results in the pumping of protons from the matrix to the intermembrane space, further enhancing the proton gradient. Now that we've covered all the stages, let's see how this proton gradient drives ATP synthesis in chemiosmosis. Chemiosmosis, the final stage of oxidative phosphorylation, where the proton gradient created by the electron transport chain drives ATP synthesis. Here's how it works. First, the electron transport chain pumps protons from the mitochondrial matrix into the intermembrane space, creating a high concentration of protons and an electrochemical gradient known as the proton motive force. Next, ATP synthase, an enzyme complex embedded in the inner mitochondrial membrane, takes advantage of this proton gradient. ATP synthase has 2 main parts: the F0 unit, which forms a channel allowing protons to flow back into the matrix, and the F1 unit, where ATP synthesis occurs. As protons flow through the F0 unit, they cause the F1 unit to rotate. This rotation drives the conversion of ADP and inorganic phosphate into ATP. Finally, the newly synthesized ATP is released into the mitochondrial matrix, ready to be used by the cell for energy-requiring processes. In summary, chemiosmosis uses the proton gradient established by the electron transport chain to power ATP synthase, producing ATP from ADP and inorganic phosphate. This process is crucial for providing energy for various cellular activities. Now, let's see how these 12 carriers produce ATP. NADH molecules enter the electron transport chain at complex 1, while FADH2 enters at complex 2. When one NADH is oxidized, it transfers electrons to complex 1. As electrons move through the electron transport chain, they drive the transport of protons across the inner mitochondrial membrane. Each NADH molecule results in the transport of 10 protons across the membrane. Since it takes 4 protons to generate 1 ATP molecule, the oxidation of 1 NADH generates approximately 2.5 ATP molecules. Similarly, 2 NADH molecules produce around 5 ATP molecules, and a total of 10 NADH molecules produce approximately 25 ATP molecules. From the 10 NADH molecules, we get approximately 25 ATP molecules. Now let's determine how many ATP molecules are produced from FADH2. As mentioned earlier, FADH2 enters the electron transport chain at complex 2, skipping complex 1. Therefore, it only pumps 6 protons across the membrane. Since 4 protons are required to generate 1 ATP molecule, the oxidation of 1 FADH2 molecule results in the production of approximately 1.5 ATP molecules. Similarly, 2 FADH2 molecules produce about 3 ATP molecules. So, by the end of the third stage, The 12 electron carriers 10 NADH and 2 FADH2 collectively produce a total of 28 ATP molecules. Now that we've covered all the ATP production from one glucose molecule, let's summarize the total ATP yield. Glycolysis produces 2 ATP molecules, Krebs cycle produces 2 GTP molecules which are equivalent to 2 ATP, and electron transport chain produces 28 ATP molecules. So, in total, one glucose molecule can produce approximately 32 ATP molecules. This comprehensive ATP yield reflects the efficiency of cellular respiration in converting glucose into usable energy. I've tried to cover the entire process in detail to make it as clear as possible. I've tried to cover the entire process of cellular respiration in detail to make it as clear as possible. Stay tuned for our next video, where we'll be diving into protein catabolism, breaking down proteins for energy. Don't miss it. If you've learned something new from this video, please hit the like button and share it with your friends. Don't forget to subscribe to Bioscholar for more content. Thanks for watching, and I'll see you in the next video.